Hello everyone, welcome to KR Microbiology classes. So today I'm going to explain about glycolysis and metabolism and related questions which were given in the 2025 and 2024 question papers. So there is compulsory question will be there, at least 2 to 3 questions regarding the metabolic activities like glycolysis, Krebs cycle or electron transport like that. So let us move to the topic. So first question is, in lactic acid fermentation, lactate dehydrogenase gene becomes non-functional due to mutation. The product that will accumulate at the end of the process is called as. So here you have to read the question again. So the formation of lactic acid from the pyruvate. This reaction was handled by the lactate dehydrogenase enzyme, right? And that regarding gene was non-functional because of mutation. So what will be the accumulating product they are asking? Here you have to simplify this question. Simply the question is, so lactic acid is not forming from the pyruvate because of there is no availability of the particular enzyme. So then what will be the accumulated product? Simply what will be it is? So it is the pyruvate, right? So they gave the options pyruvate, lactic acid, acetaldehyde, ethyl alcohol. Correct answer is the pyruvate. Okay. Next question. So I gave you some explanation also here. In the glycolysis, glucose forms as the pyruvate, and from the pyruvate to lactate, it is called as the lactic acid fermentation. Here this LDH, lactate dehydrogenase, will act for the formation of the lactate. Next question is the formation of 3-phosphoglyceric acid from 1,3-diphosphoglyceric acid in presence of phosphoglycerokinase is an example of what kind of reaction they are asking. Once again, please read the question. The formation of 3-phosphoglyceric acid from 1,3-diphosphoglyceric acid in presence of phosphoglycerokinase is an example of. Here the answers, you see that correct answer is substrate level phosphorylation. First we have to read all the 4 options, right? A. Substrate level phosphorylation. B. Oxidative phosphorylation. C. Dehydrogenation. D. Isomerization. So to answer this question, you have to know that what is the actual reaction here. 3-phosphoglyceric acid from 1,3-diphosphoglyceric acid. Here what was happened, you can assume 1,3-diphosphoglyceric acid gave 3-phosphoglyceric acid, means one phosphate was deleted or reduced, right? So please see this. Phosphorylation means here see this two, there are mainly 3 types of phosphorylations will be there. Substrate level phosphorylation, oxidative phosphorylation, photophosphorylation. The actual definition for the phosphorylation means adding of the phosphate, okay. So for this question, 1,3-bisphosphate 1,3-bisphosphoglycerate is formed, sorry, it gives 3-phosphoglycerate. Here one phosphate is given to the ADP so that it became as the ATP. This is called as the substrate level phosphorylation. Let's move see here I also gave you the glycolysis cycle. So to answer these kind of questions, first you must have to remember complete glycolysis cycle, like what are the intermediate compounds and in between which reaction ATP is forming or ADP is forming. You have to remember very particularly. Daily you have to practice this cycle daily, once or twice you have to practice, then only you will be able to remember this cycle, okay. So next we'll see the next question. In futile cycle, chemical energy is dissipated as heat due to 2 opposite biochemical reactions. Which of the following biochemical reaction is a part of futile cycle? Here the correct answer is fructose 6-phosphate, fructose 1,6-bisphosphate. What is the meaning of futile cycle? Sorry, may I pronounce it some differently, but I hope you will understood. In futile cycle, chemical energy is dissipated as heat due to 2 opposite biochemical reactions. Which of the following biochemical reactions is or are a part of futile cycle, they asked. Here first you have to understand what is futile cycle. Means the reaction which was became waste, means there is no proper utilization of the energy released. Such cycle or such reaction we will call it as a futile cycle, means there is no proper utilization and all the energy was produced became waste. So here I gave some explanations. First we will see the four options: glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glucose 6-phosphate, fructose 6-phosphate, 1,3-bisphosphoglycerate, 3-phosphoglycerate. Before you are answering this question, you must have to again remember glycolysis cycle. See here, fructose 6-phosphate becomes fructose 1,6-bisphosphate. Here 1 ATP utilized and it became as the ADP. Here 1 ATP utilized and here the enzyme participated is a phosphofructokinase 1. But here heat generated and there is no proper utilization of this particular energy. So this is the futile cycle. It will happen in the glucogenesis. Next question is, which organelle in eukaryotic cell is the site of electron transport chain? Which organelle in an eukaryotic cell is the site of electron transport chain? Very basic question, but you must have to remember all the kinds of metabolic activities, those which are happening in which part of the cell. So here see the four options. Option A, endoplasmic reticulum. Option B, Golgi apparatus. Option C, mitochondrion. Option D, peroxisome. The correct answer is the mitochondrion. Now I will try to tell you that in eukaryotes, electron transport chain will happens in the inner mitochondrial membrane, and in prokaryotes, it will happens in the plasma membrane or cell membrane, we may call it as. And you also have to remember what kind of metabolic activities are happening in which part of the cell. The major metabolic activities such as glycolysis, pentose phosphate pathway, HMP pathway, fatty acid synthesis, glycogenesis, glycogenolysis, amino acid transamination, and part of gluconeogenesis and part of heme synthesis. All these will happen in the cytosol. And if you see in the mitochondrial matrix, pyruvate oxidation, TCA cycle or Krebs cycle we may call it as, okay. TCA cycle, beta oxidation of fatty acids, ketogenesis, ketolysis, amino acid oxidative deamination, and a part of urea cycle, gluconeogenesis and heme synthesis will happen in the mitochondrial matrix. And especially in the inner mitochondrial membrane, as we discussed earlier, ETC will happen, oxidative phosphorylation will happen, and ATP synthesis will also happen. And thank you so much. Thanks for watching. I hope you will understand this class. If you have any doubts, please mention in the comment section. Thank you so much.